Section 3 in Chapter 11, especially the second half of it, really focuses on the deviations from our typical genetic outcomes. So think about the family genetics that you worked on and any of the surprises that you might have. Well, let's kind of look at Mendel's principles and see where there might be some variations. Where do those variations come from? So Mendel's principles from genetics focus on factors, genes are passed from parents to their offspring. Each parent has a pair of factors, but only one passes. Remember this idea of segregation in meiosis where those split. So each gamete gets one allele. This allele is then combined with the other parent's factor, and that's where you get the offspring's genotype. Alleles for different genes usually segregate independently, and the combination or pairing of these factors create offspring's traits, the phenotype and the genotype. And in cases where two genes determine a trait, one is going to be dominant. So those are kind of the issues or kind of the principles of Mendel, the staple for the foundation of genetics. However, there are more ways that genes can interact with one another other than just being dominant and recessive. The ones that we are going to look at for this section include incomplete dominance, codominance, multiple alleles, and polygenic traits. So let's move on to what is incomplete dominance. So incomplete dominance means that one trait is not completely dominant over another. So what's going to happen is there's going to be a blending of traits. So if we were to cross a red flower with a white flower, we'd actually get some pink flowers because they're not going to be completely, the red is not completely dominant over the white. So what we can see here, an example, so in a species, a dominant homozygous will create a red flower and a heterozygous alleles will create a pink flower and two smaller case will create a white flower. So if you cross two pink flowers, determine the possible genotypic and phenotypic ratios. Okay, so let's put these guys in here. So we have two pink. So a pink flower is going to share both. So it's going to have one dominant and one recessive, and one dominant and one recessive. So incomplete means, hey, we're going to get a combination, and they're going to be heterozygous. So in here, if I fill out my Punnett square, and again, if you are still having problems with Punnett squares, you need to come and see me because this should come pretty easy for you. So if we have two pink flowers, we're going to have RR is going to be 25%, RR is going to be 50%, and then we have our at 25%. Again, if we look at these, if they are codominant, anytime both alleles are, anytime they're heterozygous, it's gonna be that combination of the two. So we would have red, there'd be 25% red. For pink, you're gonna have 50%, and for the white, we will have 25. So this ends up being a little bit different from how we usually see the Punnett squares. When we usually see the Punnett squares, we would say, wait a minute, 3 of them have the dominant allele present, so that's what the phenotype should be, is the dominant. However, incomplete dominance tells us that, wait, anytime we have both, we are going to have a different. We're going to have more of this blending. And that's kind of the case with the curly hair. For some of you that had that issue with the curly hair, there is a blending of the traits, this incomplete dominance. So someone might have a little bit of wavy hair, and that means that they have both. So that's why some of your results for curly hair were different, because we assumed kind of the typical Mendelian genetics where there is two alleles, a dominant and a recessive. But we're starting to learn that that's not necessarily the case. So we just did this in here. We did our pink flowers. So I'm actually going to skip ahead here, and we're going to move to codominance. So codominance is when both alleles are actually going to contribute. So it's not necessarily a blending like in the one before, but you're going to actually see each of them on the organism. So one example is dealing with chickens, black and white. So a black and white rooster and a black rooster. So what happens is we have a black rooster as being crossed with a white rooster. Well, how this works is they actually create offspring that are both black and white. So they have not necessarily the dominant, we can't really say which one is specifically dominant because there is no one that wins out over the other. So that's why we notice we start to see a little bit different in how we represent that, because they're co-dominant together. So we're going to have two capital, and we're just going to change that just a little bit. So again, if we have a black and white rooster and we are going to cross it with a black rooster, this is how we're going to write it. So let me get this out of here for a minute. So if we're looking at what we had before, if the black one, so if we had something like this for the black, and our white, we are going to see a white like that. Okay, so if we cross it with just a black rooster, we are going to end it something like this. Okay, so in codominance, we're going to have a regular with a superscript. So what would we get when we actually end up crossing these? Again, the Punnett square stuff. So we're just going to kind of continue from here, and you should be able to fill this out and say, wait, what am I going to end up with? So excuse me while I take a minute to fill this out. So now that I've filled all of those out, we can have different genotypic ratios and phenotypic ratios. So we're going to end up with 50% that are going to end up like this, and the other 50% that are going to be these guys here. So again, slight variations with this. What does that actually mean? What do these end up looking like? Well, we're going to have two black, so 50% black, and we're going to have a white and black for 50%. So what this basically means is that both of these are going to express their traits at the same time. There's not going to be this blending of it. Each is going to be expressed. So on the chicken, we will see a black and a white. The third piece is multiple alleles. And this means that there's many traits, such as hair colors in rabbits, hair colors in humans, that are going to use more than a single set of alleles. Looking at just one allele doesn't give us enough information to actually figure out what the outcome would be. And it has to do with the fact that often the colors aren't clear-cut. So we can't just look at the colors and say, oh yeah, this is either black or this is brown type of thing. So when we look at the hair color in rabbits, we have a dark gray can have all of these different ones. A chinchilla can have all of these different alleles. Himalayan, albino. So sometimes there are multiple alleles that have to be involved before we can get the color. So when we look at human hair color, so like what is brown? Like if you say someone has brown hair, does that really give you an idea that what color their hair is? Not really. So we cannot simply code all of the variations for brown hair in just two different alleles. This is kind of the inheritance of brown hair color, and look at all the different shades of hair color. So that's what multiple alleles is. We need more alleles involved than just the typical two. And the last one is the polygenic traits, and this means that the combination of multiple genes determine which trait is going to be present. So a little bit different than multiple alleles. These are actually different traits, and they tend to show a wide range of phenotypes just because the trait is controlled by two genes, and remember those genes might have two alleles or more alleles, and a lot of the human traits are determined by this. So that's kind of the big thing is we start off with doing the basics and saying, hey, this is Mendelian genetics, and if each one gives one allele, we can assume dominance or recessiveness, but unfortunately that's not necessarily the case. There's 4 different ways that this can kind of go a little bit off. So we have our incomplete dominance, codominance, multiple alleles, and polygenic traits. That's it for this section.